Very good evening to all the NEET aspirants 2026. This is your Prasanna ma'am from NEET World. So, in today's session, we are going to have a detailed discussion on cell wall and cell membrane, which are the limiting boundaries of plant cell and animal cell. So, now first if we have to see cell wall formation. First, let us look into the composition of cell wall. In what organisms can you see the cell wall? Cell wall is not just present in the plant cell. Cell wall is observed in a fungal cell, algal cell and a bacterial cell as well. First, let us look into the composition of the plant cell wall, algal cell wall, bacterial cell wall and fungal cell wall. Plant cell wall is composed of what ma? Cellulose, hemicellulose, pectin, cellulose, hemicellulose and pectins. This is the composition of plant cell wall. Algal cell wall, okay. Algal cell wall has mannans, galactans, calcium pectate and calcium carbonate, okay, calcium carbonate, okay. Then fungal cell wall. Fungal cell wall is made up of what? Chitin. It has chitinaceous cell wall. Bacterial cell wall has what ma? Peptidoglycan. So, here when you can see the compositions of the cell walls, I would like to give you one point from Linnaeus two kingdom system of classification, where in that two kingdom system of classification, the main criteria concerned is cell wall, because of which fungi, bacteria, algae, plant cell, everything are placed only in the one kingdom, that is kingdom Plantae, okay, and just look into the compositions. Now, let us see the structure of plant cell wall, okay, the structure of plant cell wall. First, during the time of cytokinesis time, how cell wall is formed, let us observe. After telophase stage in the plant cell, what happens is, after telophase stage, if this is the plant cell, two nuclei are present, fine. So, here at the center what happens is, first phragmoplast is formed. What is formed ma? Phragmoplast. This is called as phragmoplast. This is being secreted from the organelle, that is Golgi bodies. It is being secreted from Golgi bodies. Fine. Golgi bodies will synthesize phragmoplast. This phragmoplast will eventually develop to cell plate, okay, by moving from center to periphery. This phragmoplast will extend from center to periphery. The movement from center to periphery, this type of movement is called as what ma? This is called as centrifugal movement, centrifugal, okay. So, because of this centrifugal movement, initially cell plate is formed, cell plate. This cell plate will completely bind and it will mature into the middle lamellae. The cell plate will mature into what ma? Middle lamellae. It is maturing to middle lamellae. Clear beta. So, here first initially what is produced? Phragmoplast. Phragmoplast is being synthesized from Golgi bodies. This eventually grows from center to the periphery. This is centrifugal movement and this forms a cell plate and middle lamella is formed at the end. So, middle lamella is the first formed layer of the cell wall in a plant cell. What is first formed? Middle lamella is the first formed layer, okay. Next to the middle lamella, inner to the middle lamella, primary cell wall is formed. Okay, inner to the middle lamella, what is formed, ma? Primary cell wall is formed. Now let us suppose this is the middle lamella which is formed in between two plant cells. Okay, this is the middle lamella, first formed layer in a plant cell, and this is the plant cell in between. Okay, this is the cell membrane of the plant cell, let us suppose. Next, inner to the middle lamella, primary cell wall is formed. Inner to the middle lamella, what is formed, ma? Primary cell wall is formed. This is middle lamellae and this is primary cell wall. Primary cell wall is thin, it is thin, elastic, and it is easily extensible and it will stretch. Fine. So, this is about the primary cell wall. Primary cell wall has usually made up of cellulose. Fine. Cellulose and hemicellulose. Primary cell wall is made up of cellulose and hemicellulose. Next, inner to the primary cell wall, what is formed next to the primary cell wall? Inner to the primary cell wall, secondary cell wall is formed. This secondary cell wall is 2 to 3 layered. Secondary cell wall is 2 to 3 layered. This has thick and tough lignin suberin depositions. So, next, inner to the primary cell wall, what is formed, ma? Secondary cell wall. This has thick lignin and suberin depositions. Suberin depositions. Clear. So, here middle lamellae will be having what? Calcium pectate and magnesium pectate. Calcium pectate and magnesium pectate. Compositions are very important, beta. So, here if you can see, from outer to inner side, first formed layer is middle lamellae. These are made up of calcium pectate and magnesium pectate. That is why in ripened fruits, if you see, over the cell walls, this middle lamellae gets dismantled, and that calcium pectate and magnesium pectate will give that brittle nature to the fruit pulp. Okay, that is about middle lamella, first formed layer of the cell wall. Inner to the middle lamella, primary cell wall is present. This is thin, elastic, extensible, and this is made up of cellulose and hemicellulose. Next to the primary cell wall, 2 to 3 layered, what is present? Secondary cell wall is present. Secondary cell wall is tough and thick because it is having lignin suberin depositions inside it. As the cell grows and as the secondary cell walls are formed, primary cell wall will get degenerated. Okay, primary cell wall will get degenerated as the secondary cell wall layers are formed and growing. Now, you tell me, cell wall is rigid and it is dead layer, right? Cell wall is rigid and dead. There is one living tissue which is present in the cell wall, that is plasmodesmata. Okay, the only living cell present here is what? Plasmodesmata. Plasmodesmata connections are in between two cytoplasms. Okay, this connects the cytoplasm of two plant cells. Plasmodesmata will connect the cytoplasm of two cells. Cytoplasm of two cells. This plasmodesmata, inside it has desmotubules. Plasmodesmata will have desmotubules. Clear, ma? Clear with the cell wall structure and different cell wall compositions. So, if at all you are getting a question, which layer is close to middle lamella? The layer which is close to middle lamella is primary cell wall. Which layer is close to plasma membrane? The layer which is close to plasma membrane is secondary cell wall. Now, let us plot out from plasma membrane to middle lamellae. First, plasma membrane, And next, closest layer to plasma membrane is what? Secondary cell wall. Next, after secondary cell wall, it is having primary cell wall, and the last layer is middle lamellae. Okay, and the last layer is what, ma? Middle lamellae. Clear? So, plasma membrane, next it is primary cell wall, secondary cell wall and middle lamellae. From outer to inner, if you see the sequence of the cell wall layers in plant cell, it is middle lamellae, primary cell wall, secondary cell wall and plasma membrane. Remember that properly so that any question in any form you may get also you can answer. This is the complete description of the cell wall, how the cell wall is being formed and how it is being growing and what is the living cell, living tissue between two in the cell wall which is connecting two cytoplasms, where this is cytoplasm 1 and this is cytoplasm 2. Plasmodesmata is connecting. Plasmodesmata is having what protein lines beta? Desmotubules. So, this is all about cell wall of plant cell. Now, let us go to the limiting boundary of an animal cell, that is cell membrane. In the year 1950, because of the evident development in electron microscope, okay, electron microscope, cell membrane is being studied and there are many models proposed for cell membrane. But the model which is widely accepted is Fluid Mosaic Model, which is given in the year 1972 by Singer and Nicholson. Okay. So, the Fluid Mosaic Model is given in the year 1972. That Fluid Mosaic Model is widely accepted, and this is proposed by whom? Singer and Nicholson. Now, look into the composition of the plasma membrane. Plasma membrane is composed of what? Okay, plasma membrane and Singer and Nicholson has proposed the cell wall structure or cell membrane structure, that is Fluid Mosaic Model, observing the cell membrane of erythrocyte or RBC. In erythrocyte or RBC, what will be the protein and lipid composition? Lipid composition is 40%, lipid composition is 40%, whereas protein composition is 52%. Protein composition is 52% and lipid composition is 40%. This is about cell membrane. So, here you see first plasma membrane, it is having three components inside it. What are they? Lipids, proteins and carbohydrates. Lipids, proteins and carbohydrates. How lipids are composed of? What type of lipids are composed of? In Fluid Mosaic Model, you can clearly see the lipid bilayer. In lipid bilayer, you can see these structures. Okay, these are the structures of lipid bilayer, wherein you can observe the hydrophilic head, which is polar. This is polar hydrophilic head, which is projecting towards the aqueous environment outside. Okay, this is towards the aqueous environment. And inner side you can observe hydrophilic tail. Okay, this is hydrophobic tail. Hydrophobic tail is non-polar. So, if there is a statement that is being asked, the non-polar hydrophobic tail is protected from the aqueous environment by hydrophilic polar head. Clear? So, this is about lipid bilayer. Fine. And how the lipid bilayer is arranged in the plasma membrane? It is of liquid nature, quasi-fluid nature will be the nature of lipid. Now, what types of proteins can you see inside the plasma membrane? You can observe three types of proteins: peripheral proteins, integral proteins. Integral proteins are also called as tunnel proteins. Integral proteins are also called as tunnel proteins. And third type will be transmembrane proteins. Transmembrane proteins is third type. Okay. So, you have peripheral proteins, integral proteins or tunnel proteins, and transmembrane proteins. Peripheral proteins will be on the peripheral surfaces of the plasma membrane. Integral proteins will be inside the plasma membrane, transpanning two sides of the lipid bilayer. Okay. Transmembrane proteins can transpan any one of the sides of the plasma membrane. Okay. Transmembrane protein will transpan any one of the sides of plasma membrane. So, this is about integral proteins, peripheral proteins and transmembrane proteins. Clear ma? So, now there is one popular statement that all integral proteins are transmembrane proteins, but not all transmembrane proteins are integral proteins. Okay. Because all integral proteins will transpan two sides of the membrane, but transmembrane proteins will either on the one side or two sides sometimes. Okay. So, all integral proteins are transmembrane proteins. Clear ma? Next, carbohydrates. What type of carbohydrates are observed and where are they present? Okay. Carbohydrates are just 5 to 8%. How they are present? They are present in the form of glycolipids and glycoproteins. They are present in the form of glycolipids and glycoproteins. Okay. So, glucose units or sugar units attaching to the lipid layer, it is glycolipids. If they are into protein layer, it is glycoproteins. Clear ma? So, this is the composition: lipid, protein and carbohydrates. Hydrophilic head is polar, that is towards the aqueous environment. Hydrophobic tail is non-polar, that is inside the lipid membrane, lipid bilayer. Proteins, peripheral proteins, integral or tunnel proteins and transmembrane proteins. Carbohydrates are in the form of glycolipids and glycoproteins. These are secreted from Golgi bodies. Okay, glycolipids and glycoproteins are secreted from Golgi bodies. Clear ma? Now you see, there are few statements from your NCERT which are very important for your NEET. Okay. So, first thing, proteins can move in the lipid bilayer. How? The quasi-fluid nature of lipid will enable the lateral movement of proteins. Okay. The quasi-fluid nature of lipids. Quasi-fluid nature of lipids will enable the lateral movement of proteins. Lateral movement of proteins. Clear? Quasi-fluid nature of lipids will enable lateral movement of proteins. What does it mean and what type of movement you can observe? So, in the lipid bilayer, if this is the lipid bilayer, inside this you can see the proteins are of many types as we have discussed: peripheral proteins, integral proteins or transmembrane proteins. Protein movement within the lipid layer, only single layer, monolayer movement is happening, where proteins can be moved from one place to other place. This is called as lateral movement. Lateral movement of proteins. Fine. And the proteins are moved from one lipid bilayer to the other. This is called as flip-flop movement. Okay. Flip-flop movement. So, lateral movement is possible both for lipids and proteins, but flip-flop movement is only seen in proteins. Clear? This is possible because of only one thing, that is quasi-fluid nature of lipids. And there is one more important statement here, that how proteins are seen on the lipids. Proteins are present as icebergs on the sea of phospholipids. What is the major lipid content in the plasma membrane? Phospholipid. Okay, phospholipid is the major lipid content in the plasma membrane. So, how the proteins are present in the plasma membrane? Proteins are present as icebergs in the sea of phospholipids. Take that statement, most important mark. Okay, proteins are present as, how proteins are present? Proteins are present as icebergs in the sea of lipids, in the sea of phospholipid bilayer. Phospholipid bilayer. Clear, ma? Understood, right? So, this is the important term about plasma membrane. Now, you have completely understood the composition, the structure, the movement in the plasma membrane. Now, let us look into the functions of the cell membrane. And I would like to add on here two more terms. Cell membrane, you all know, it is also called as plasma membrane. It is also called as semi-permeable membrane. It is also called as selectively permeable membrane. Why is it called as semi-permeable membrane? Why is it called as semi-permeable membrane? When plasma membrane is only allowing the solvent molecules, but it is not allowing the solute molecules, then it is called as semi-permeable membrane. Selectively permeable membrane. Selectively permeable, which means that in the solute particles or ion channels also, it is selectively permitting the solute particles, then it is called as selectively permeable membrane. Look into the synonyms properly. So, we have clearly understood the structure and composition. Now, let us look into the transport or the functions. The major functions of the plasma membrane includes cell growth, formation of intercellular junctions, secretion, endocytosis and cell division. These are the major functions that are given in our NCERT, but of which transport is very much important. How the transport is being taking place. Listen here properly, ma. Transport is of two types. Okay, transport, plasma membrane transport is of three types actually. I will tell you. First is passive transport. Second is active transport. And third will be bulk transport. Third is what, ma? Bulk transport. Fine. So, you have active transport, passive transport and bulk transport. What is passive transport? The movement of molecules without any requirement of energy is called as passive transport. What comes under passive transport? Okay, diffusion. Okay, what comes under passive transport? Diffusion, facilitated diffusion, facilitated diffusion and osmosis. These are the three patterns of passive transport. Okay, what are the beta? Diffusion, facilitated diffusion and osmosis. Let us look into all the three types. So, here you can see diffusion, you all know. Normal neutral solutes and gaseous molecules, they transport via diffusion. Okay, just from high concentration to low concentration, gaseous molecules might be, neutral solutes might be, they will be diffused from one cell to other cell through the membrane, inside the membrane, they will just diffuse from one cell to the other cell. Okay. So, that diffusion or the movement of the molecules, be it neutral solutes or gaseous molecules from high concentration to low concentration is called as what? Diffusion. Second is what? Facilitated diffusion. Facilitated diffusion is nothing but the ions, few ions and few molecules, especially polar molecules like water molecules. Transport of the water molecules or any ions from high concentration to low concentration via carrier proteins or channel proteins. Okay, it is not by using energy, but via carrier proteins or channel proteins is called as what? Facilitated diffusion. And osmosis, you all know that movement of molecules from high concentration to low concentration through the semi-permeable membrane is called as osmosis. Water is being transported either via facilitated diffusion or through osmosis. So, now you can, I hope you have understood clearly. For diffusion, what are the molecules that are getting diffused? The very first thing is what ma? Gaseous molecules, and neutral solutes. Neutral solutes will diffuse from cell to cell. Next, facilitated diffusion. Facilitated diffusion is via what? Carrier proteins or channel proteins. Carrier proteins or channel proteins. Osmosis. Osmosis is movement of molecules from high concentration to low concentration through plasma membrane or semi-permeable membrane. Okay, this is called as osmosis. This is all about passive transport. Active transport is specially for ion channels like sodium potassium pumps. Okay, sodium potassium channel, wherein sodium is moving out of the cell, potassium towards inner side, like that sodium potassium pumps. Anything might be movement of molecules or ions by using ATP or energy, okay, which requires energy requirement, that is ATP. Okay, the transport which requires energy or ATP is called as active transport. Okay, here you can see the molecules moving from low concentration to high concentration. As the movement of molecules is from low concentration to high concentration, this requires energy, that is ATP. So, clear ma? Movement of molecules from low concentration to high concentration here. This is from low concentration to high concentration. Fine. This is from low concentration to high concentration. In diffusion, facilitated diffusion, osmosis, this is from high concentration to low concentration. Clear, right? This is from low to high. Clear? Now, bulk transport. Bulk transport is nothing but movement of complete molecules, so many molecules either into the cell or out of the cell, that is bulk transport. In bulk transport, if you have to see, two types of bulk transport is observed. One is endocytosis, and the other is exocytosis, endocytosis and exocytosis. Endocytosis, if it is engulfing water molecules, any lipid droplets or water molecules, that is called as penocytosis. Penocytosis, if it is engulfing or taking inside lipid molecules or water droplets, that is called as cell drinking. It is called as what ma? Cell drinking. Fine. This is penocytosis. Second is phagocytosis. Okay, phagocytosis, where cell will engulf or it will take the solid molecules, solid particles, that is called as cell eating. Okay, cell eating. And whichever toxin molecules which are present after lysosomal action or the cellular constituents which are toxic inside the cell or some dead organelles inside the cell, they are omitted out of the cell membrane, that is called as exocytosis. Exocytosis is also called as effaggy. Okay, effaggy or cell vomiting, it is also called as cell vomiting. Clear ma? So, this is all about the transport. Passive transport, which is from high concentration to low concentration. Diffusion, gaseous molecules and neutral solutes will pass by our diffusion. Facilitated diffusion is mediated by carrier proteins and channel proteins. Osmosis, is from high concentration to low concentration through the membrane. Active transport is from low concentration to high concentration, which requires ATP energy. Best example is sodium potassium pump. Third mode of transport, that is bulk transport. It can be of two types, endocytosis and exocytosis. Endocytosis, best example is pinocytosis, that is cell drinking, and phagocytosis, which is cell eating. Okay. Exocytosis, which is also called as effagi, where it eliminates the toxin molecules or the dead constituents which are in the cell. This is also called as cell vomiting. So, this is all about the cell membrane beta. So, this is all about cell membrane and these are the major functions of plasma membrane. I hope the complete information of cell wall and plasma membrane is clear to you. In our next session, we are going to deal with endomembrane system. From that concept of cell, the unit of life, definitely you will get a question. So, don't miss the coming session, that is endomembrane system. Thank you.